is about the cell membrane. It's the next section of chapter 2. We've talked about macromolecules, and now we're going to talk about the cell membrane, how its structure and how the macromolecules relate to its structure too. There's a little picture there on the right about the cell membrane. We're going to actually draw it so you can see all the different components in a little more detail. And there are three main kind of phrases they use to describe the cell membrane. The first is that it's selectively permeable. So if you think about what permeable means that things can pass through. Okay, so and then if we put selectively in front, selectively permeable means, oh, well, only certain things are going to be allowed to go in or go out, to pass through, basically. So the cell membrane is selectively permeable, and that's important because it controls what can go in or out of the cell. The second phrase is that it's a phospholipid bilayer. Okay, so if you think, let's look at the word bilayer. Bi means two. So it is made up of two layers of these molecules. So 2 layers of phospholipids. If you look at the diagram, here's one layer of phospholipids, and here's the other layer of phospholipids. And they arrange themselves that way because of their tendency toward water. Okay, so this is just a schematic diagram of a phospholipid. So we're going to take a look at a phospholipid. And the phospholipid has a phosphate group in the head, so at one end. Okay, so the circle represents the phosphate head. And it's slightly charged, and so it interacts well with water. It's said to be hydrophilic. So that's why the heads are arranged here, because then they kind of protect the hydrophobic parts against water. Because right here, we have cytoplasm, which is mostly water, intercellular fluid, which is mostly water, and this is the outside of the cell. And outside of the cell, up here, is mostly water as well. So there's a lot of water on either side of the cell membrane. So the polar heads of the phospholipids will arrange themselves so that they are facing the watery surroundings. These tails are really fatty acid tails, and the fatty acids are hydrophobic. Okay, we've seen that before. The reason it's called phospholipid, lipid is, well, they're a class of molecules that don't interact well with water. They are hydrophobic. Okay, so this long chain, this fatty acid tail, is a long hydrocarbon tail, and they don't interact well with water, and so in order to protect themselves from water, they orient themselves towards each other on the inside of the phospholipid bilayer. Okay, so the tails go on the inside, and the heads, tails go on the inside, and the heads are on the outside towards the water. Okay, so this is really important so that you can be able to explain why it forms the phospholipid bilayer because of the components of the phospholipid and whether they like to be near water or not. If you remember how oils interact, if you were making a pot of spaghetti, for instance, and you were getting the water ready and you put a little bit of oil in it, and you look at what happens with the oil. Oil doesn't like water, so you end up with these little droplets that sometimes will kind of coalesce, or they kind of come together and form a bigger droplet, but they're rounded because they're trying to hide their hydrophobic parts from the water molecules. Okay? So the same kind of thing is happening here to form this boundary around the outside of the cell membrane. So not only are there phospholipids in the membrane, so there's lots and lots of phospholipids. It's mostly made up of phospholipids, but there's a lot of things embedded in the membrane. Okay, a lot of proteins, and these are all different proteins. There's one kind, another. We're going to do a little drawing that shows a lot of different types. Now, what types are on a given cell depends on what that cell does. If the need for that cell to bring in glucose, then they're going to put out certain transport proteins or channels to help get that glucose through the membrane. Okay, so it depends what the cell is doing, what kind of a cell it is, what its main job is, what kind of proteins it would have embedded in it. There's also some cholesterol. It's not shown here, but we could kind of put some in here. We can't see that very well. So red. Okay, so there's some cholesterol molecules, and we're just going to represent them with these little red things. And the cholesterol is embedded in there to give it rigidity. So the more cholesterol that's there, the more rigid that region of the membrane. You might benefit from going back and watching that video from the last chapter that showed the cell and the things interacting inside of the cell, because you might pay more attention now to the membrane itself and watch as things are kind of flowing through. There's like little rafts that can pass along through the membrane because of its structure. So when you talked about the lipids and the structure of fatty acid tails, we learned that if they're an unsaturated fatty acid, then it introduces a kink. Okay, so the fatty acid tail wouldn't just be straight then, you'd have a kink. And those kinks are really important because we talked about how if they're all saturated, like in butter for instance, if all of those fatty acid tails are saturated, then they can really pack close together and make like a solid. But in the cell membrane, we have a lot of these unsaturated fatty acids, so there's a lot of kinks introduced in these phospholipids, and because there's so many kinks, they can't pack really, really tightly together, and in fact, it's more of like a freely moving, continuously moving membrane, in that things can kind of pass along or through or pass around along the membrane, as long as it's compatible with the structures that are inside. So, if you remember from chemistry class, they say like dissolves like. Okay, so things that are more hydrophobic and not too big should be able to pass easily through the hydrophobic region that is this big boundary here. Okay, so this membrane right in here, all this stuff is hydrophobic. Okay, so if we talked about certain hormones, some of them are lipid-based, so they would be able to pass right into the cell, and they have their action right in the nucleus of the cell, in fact. Okay, so things like ones that act like testosterone, so steroids that people take, actually will pass right into a cell and go into the nucleus and affect production of proteins and other things in your body. Whereas other types of hormones, like protein hormones, they're not hydrophobic and they can't get through the cell membrane, so they have a different kind of action where they have to actually bind to a receptor on the inside. Now, so that kind of brings us to the third point here. This is another phrase that they use to describe the cell membrane. They say it's a fluid mosaic. Oh, there goes the bell. A fluid mosaic. And so fluid just is referring to the fact that because of all those kinks in the fatty acid tails, the membrane is not a solid, rigid structure. Okay, it actually allows things to kind of pass along or pass through at times, depending on their nature. And it's a mosaic. If you think of an art piece that's a mosaic, it means that there's a lot of different things in it. So if you look at the cell membrane, at any time there can be a lot of different types of proteins embedded in it, some cholesterol and some other things, So it's kind of like a mosaic, or has a lot of different pieces or different things embedded in it. So we're going to take a little bit to draw a cell membrane. Okay, and I may end up cutting this part out if it takes too long, but I think it's important, and it's so important that I give time in class to draw it with me. So I'm going to draw, and I'm not the most artistic, so draw. Well, I don't think I need quite that many to start out with, but it's alright. You don't have to have the exact same number of phospholipids. Alright, probably easiest if you draw this with your paper going, not portrait, but landscape, so that you don't get frustrated if it goes too long for you. It's a rather long piece, can go all the way across the page. I'm going to put 6 things embedded in it. Alright, so there's the first one. Leave some space at the top and underneath. And the heads and tails should really be attached. Oh, that's too many. We'll run out of space. Alright, so another one. Let's see. Let's do this. Okay. Alright, so we're going to do blue. Again, the colors aren't that important, but it helps if they're different, just to make them stand out a little better. Another one that could go all the way through, or it might only be part way for that type. Now, this one we're drawing in 2 kind of versions. One where it's open in that direction. Not the best, but you'll get the idea. Alright, so with any drawing, it's really important that you give the viewer, pretend like they don't know anything, so you kind of have to give them an idea, okay, what is it that we're looking at here? What's up there? What's down there? What are those different colored things? What are you talking about? So, let's say we have a cell. Okay, this one is big. Let's say we have a cell. So we're going to take a piece of the cell membrane and we're going to blow it up. That's what we've done. Okay, so we need to say, okay, well, what side of the cell is what on? You know? So I'm going to say, this is the extracellular fluid right here, or space, you could say. And then inside of the cell, what's inside the cell? Well, we could say intracellular space, or intracellular fluid. Oh, another name for the intracellular fluid, we could have called it cytoplasm. Okay, so either one's fine there. So this is inside of the cell, and then up the top is outside of the cell. We're only drawing like one little section. Now, those black things are really plentiful. So let's assume that they don't know anything about those phospholipids. So you could put a box around here, and you phospholipid bilayer. I don't have room right now, but you could do that. You could also, important to kind of say, oh, what's this thing? Well, this is a phospholipid. Well, if I'm going to describe it, then I want to talk about, oh, well, this is head, phosphate head of the phospholipid, and this is a fast fatty acid tail of the phospholipid. I can't talk and write at the same time. Fatty acid tail of the phospholipid. So that when I talk about it, when I'm talking about the membrane structure, which is really, really important, by the way, then I've got it all so that people can look and say, oh, that's what that is. I get it now. Alright, so you would talk about why they're arranged that way, why they are in a phospholipid bilayer, all that detail that we already just kind of went over. So, now we need to label these things that are in here. So these are talking about 5, the general types of proteins that you might find embedded. The reason that I can tell that way up top there is the outside of the cell membrane is because of this little green thing that's sticking up over here. So this is a sugar group that sticks out, and we refer to it as glyco. Okay, and the part that's embedded in the membrane is a protein, so we call that one a glycoprotein. Glyco because it has a sugar part sticking out, protein because it is a protein embedded in it. So that sugar part that's sticking out there is important for identifying this cell. Okay, it allows other cells to kind of look and say, hey, I know what that is. Or, hey, that belongs to me. That's a self thing. I don't want to attack it. Okay, so it's important for cell-to-cell recognition and identification. The second one, okay, this kind of looks like it has a door at the top. So that one in your book is often called a gatekeeper protein. It's a protein channel, gated protein channel. And those can be triggered, so it might be a voltage-gated channel, so that would be triggered by a charge, like when in your nervous system. If a charge comes along, that might trigger that channel to open and allow certain things to pass through. Or it might be what they call a ligand-gated channel, so in that case, some certain thing has to bind to it in order to open that little door or allow other stuff through. Now, the third one, the blue one, well, it needs something of that special shape to go and bind to it. And when it does, it causes a series of reactions inside of the cell to kind of change its activity. So, this is a receptor. Okay? So, and that green thing on the outside is likely a hormone. So hormones will bind to a receptor and then they cause a series of reactions to happen inside the cell for whatever needs to be happening at the time. The fourth one, again, I said it could go all the way across the membrane or it might be just stuck in a certain part. And sometimes they're even just floating around inside the cell. This is an enzyme. Enzymes are basically, they're biological catalysts. Their job is to speed up reactions. Now the last two are really showing two versions of one kind of protein. So this is a transport protein. Now we can have transport pumps, we can have capture and release kind of transport proteins, or we can have facilitated channels where we have facilitated diffusion where there's just a channel that kind of lets it pass through. But this one is kind of showing the type where it's specific for a certain shape. So this one wouldn't fit it. This one would fit it. And it causes a change in the conformation of the protein, and it would allow it to be released on the other side. Okay? see the descriptions of those in your notes. Okay, so those are the notes that go along with talking about the membrane. Oh, but we did miss drawing one thing that should be there. Let's see. One thing that should be there, we already kind of drew it on the other page. We're going to draw them here too. Okay, and we're just going to represent them with little red squiggles. This is cholesterol. Remember? Cholesterol. And cholesterol's job in the membrane is to alter the rigidity. So if there's more cholesterol there, then it's more rigid. If there's less cholesterol, then it's more fluid, able to have movement wise in that region. Okay, so that is it for the cell membrane itself. Next day we're going to be talking about, or the next video will be about the different types of transport. There are two main ones. We're going to look at what kind of overall characteristics those two main kinds have, and then we're going to look at some examples of each.